In our previous study, we looked at phylogeography with regards to haplotype network analysis. In this video, we are going to be focusing on the correlation between phylogenetic trees and geographic location. More specifically, we are going to use the sample of looking at Lake Sturgeon, Acospencer fruvescens. This organism is one that is near and dear to my heart, as this is the organism that I used when conducting research at RIT during my student years. This particular group of organisms is extremely fascinating, as they date all the way back in existence to the lower Jurassic period and can even be found around the area of study during modern times. As we look at these particular organisms, we want to think about what their current home range is, thus we can compare it to previous history. As we look at their current home range, they are found in the tributaries of the Mississippi River and then migrating up into the Great Lakes Basin and the drainages within them. Thus, they can be found throughout North America in the mid to eastern United States and up into southern areas of Canada. This particular study focused on the area closer to home, looking at the Great Lakes Basin and some of the tributaries migrating into Ontario, Canada. It was hypothesized during this study that there would be a low genetic diversity of the specimens collected in this area due to the formation of the lakes in what would be considered, quote unquote, recent geographic time. As this study was performed, 42 samples were collected within the Great Lakes, primarily fin clips, but also some muscle tissue as well. Similar experimentation to what you performed prior to spring break was conducted, where I looked at performing DNA extraction followed by nanodrop to check quantity and quality of the DNA samples. PCR were ran, followed by a second nanodrop, gel electrophoresis, and eventually DNA sequencing. In this, the particular loci of interest was mitochondrial cytochrome B, very similarly to the size fragment that you observed during your genetic analysis during last Thursday's lecture. As we look at the provided sequence, we should note that there are 3 distinct haplotypes that are shown. First and foremost, I want you to pay attention that all 42 samples are not provided. The reason for this is the LNR16, the Lower Niagara River sample, is the representative sample of 39 different specimens, all residing within the main bodies of the Great Lakes waters. This is where we found the most prominent haplotype across all of the samples being studied. A second and third haplotype were found in other geographic locations. Haplotype number 2, represented by our MRC 05 and 01, represent haplotype number 2, being comprised of those organisms found in the Matmagami River in Ontario, Canada, one of the tributaries of the Great Lakes. Our third haplotype of interest, represented by SLR05, corresponds with those specimens found within the St. Lawrence River. It should also be noticed that on here there is a 5th specimen. This is a paddlefish. This DNA sequence was obtained from NCBI's GenBank, just the same way that you did last Thursday. The idea here is that I obtained this particular sample to be utilized as an outgroup, as paddlefish are distantly related to our sturgeon species. Now, as we look at each one of these sequences, things that are important to see from our findings but are not all depicted on this visual imagery is we noticed that there were very minimal base pair differences between all 42 samples. In this case, we noticed that those organisms residing in the Matmagami River in Ontario, Canada, exhibited a single base pair difference, where those residing in the St. Lawrence River had 2. Just like we talked about with our haplotype analysis, it happens that all of these base pair differences were synonymous, meaning they resided in the 3rd base pair position, resulting in no amino acid change. That is something common that we see occurring in cytochrome B mitochondrial DNA sequences. Now, as we think about this, let's put this data into a phylogenetic tree and see what our results are. As we look at our phylogenetic tree, you should notice that two trees are present, one parsimony and one maximum likelihood. The idea here, if you remember, displaying both of these trees based on the aforementioned genetic information gives us an idea of looking at parsimony, where the data will be explained with the least amount of evolutionary change, meaning we have the least amount of branch lengths that are occurring in this particular tree. It should also be noted that in parsimony, all the trees at speciation events are roughly the same length. There's no difference with regards to that. In comparison to maximum likelihood, which is a character-based method that incorporates the length of the branches of the tree in correspondence with the likelihood that there's a correct representation of those phylogenetic relationships within those species. Now, as we look at these relationships a little bit more, one thing we should notice is that looking at the outgroup, we have one divergence that results, regardless of parsimony versus maximum likelihood, one monophyletic clade that shows the relation of all of the Lake Sturgeon in this particular study. But one should also note that when observing our haplotypes 1, 2, and 3, we should also notice that there is another divergence that is occurring. We can see that our haplotype number 3 in this example, with MRC01 representing those organisms within the Matmagami River, has been separated out and providing the samples in the lower Niagara River and the rest of the Great Lakes Basin, as well as the St. Lawrence River, creating a sister taxa in their own monophyletic clade. If we look at the parsimony tree, we can see that according to that, the relationship is fairly confident, giving us 100% bootstrap value. But the same cannot be said with regards to the maximum likelihood tree, as we can see here looking at a 65% confidence with regards to the relationship between the majority of the samples studied and those within the Great Lakes Basin, though it still supports this idea that all of the lake sturgeon that are studied are in a single monophyletic clade. One may think, why is that occurring the way it is? Well, to understand that, some may suggest that this is a potential example of allopatric speciation, where genetic differences are occurring over a prolonged period of time due to geographic isolation. Let's see if that's true by looking at those home ranges once again, as well as looking at a little bit of historical geography. Once again, if we observe those home ranges, we're focusing on the Great Lakes Basin and the tributaries up into Canada. Currently, based on our genetic differences, it shows that we have one haplotype located up here in Canada, the majority of the organisms residing in haplotypes throughout the Great Lakes region, and our third haplotype residing in upstate New York in the St. Lawrence River. Why would we see such differences across those? Well, let's take a deeper look into the history of this particular area. One may suggest that modern demographic and ecological characteristics are consistent among the Great Lakes. Well, that may be said true today. That was not always the case. As we look at these two images, these show us a little bit more about the historical relationship between the geography in the area and how that potentially correlates with the genetic diversity that we are seeing with these particular species. As we observe this, one of the things I want you to note is that we are looking at overall glacial movement, focusing on the movement of the Laurentine ice sheet across northeastern and central North America. As we see the movement of this sheet, initially we see the formation of what is referred to as Lake Agassiz. The formation of this large body of water resulted in a divide between the Hudson Bay in Canada and the Mississippi River in the United States. It is suggested that this potential divide in a prolonged period of time could have resulted in why those samples found in the Matmagami River, tributary of the Great Lakes in Ontario, Canada, show genetic diversity in comparison to the rest of the organisms found within what will become the Great Lakes Basin. Now, as we can see here in this diagram, the light blue is representing the initial square footage of the Lake Agassiz, and the dark blue representing the modern lakes that are found in that particular area of Canada. If we continue to look at how that glacial lake or that glacial ice sheet moved, we can see that as it moves across, starting at roughly 14,000 years ago and moving to 4,000 years ago in more recent history, this is where we see the formation of the Great Lakes Basin. Now, as this ice sheet continued to retreat, one of the things that's important to note, besides obvious geographic and geologic changes, there were also ecological changes to the area as well. Thinking about things like temperature changes, water level changes, and then the potential formation of other things that weren't normally present in that area, like heightened land masses. This could be the potential suggestion when we are looking at this particular area right here. As the glacial movement occurred, we see the formation of what we now know as Niagara Falls, causing a huge barrier because of a large landmass, limiting migration between organisms above and below the falls. This may suggest that this is why we have genetic diversity between some of the organisms in the Niagara River in comparison to the St. Lawrence River, because of the presence of that huge migration barrier. It may also suggest that looking at this, even though 4,000 years ago seems extremely long to us, that's not very long when we think about geologic and geographic history of the world and Earth. This may suggest, thinking about this time frame, a very short geologic amount of time ago, that this is why we have low genetic diversity in these particular organisms, as this is resulting in what many would consider genetically and geographically recent colonization in that particular area. Thus, it can be concluded that due to glacial movement, it suggests some very plausible reasons as to why we see three distinct haplotypes, one of those falling in Canada, one of those falling in the majority of the Great Lakes Basin, and the other falling in the St. Lawrence River, essentially opposite areas of the home range stretching from north to south and west to east with regards to the Great Lakes Basin. Now, at the end of this particular study, I was curious if other lake sturgeon across the globe corresponded with my findings of just the lake sturgeon in North America, focusing on, yet again, those in the Great Lakes Basin. Thus, I moved to the next section of my particular study. In the second portion of the study, I utilized my data along with several samples from NCBI's GenBank. More specifically, I collected 24 of the 26 species of family Asperidae to see how they related to one another. As currently during the time of my research, there was no speculation as to how all of the sturgeon species across the globe were genetically related to one another, particularly in focusing on the mitochondrial B, cytochrome B loci. As we look at all of the results from our NCBI, I once again produced both the parsimony and maximum likelihood tree that you can see right here. What I encourage you to do is look at these particular trees and see what kind of things that you can find. Please note that as we look at this, the initial three haplotypes that are found in the previous portion of the study are located in one particular clade in comparison to the rest of the organisms. Hence, these are the representative of the lake sturgeon species within all of the sturgeon. As we look at the genetic differences between the trees, remember to think about branch length and the correspondence of the bootstrap values to determine the confidence intervals and how species are related to one another, as this is something as fascinating. Now, one of the things I want you to know is looking at these particular trees, focusing primarily on the maximum likelihood, as one would suggest that there's not a lot of difference with regards to the branch lengths. I want you to think about what that means with regards to the genetic differences between these very different organisms across the globe. After all, sturgeon species have been known to be found in subtropic, temperate, and subarctic rivers and lakes all across the globe, primarily focusing on North America and Eurasia, and even found in areas of the Atlantic as well as the Pacific Oceans. Thus, as we bring this video to an end, I want you to take a look at these two trees. What you are observing is the previous maximum likelihood that has been divided into two major distinctions. one separating out what we refer to as the Atlantic clade, and the other separating out a second and a third clade, the Pacific and the Stereo-Oxyoclade. These are very interesting, and what I would like you to do is I want you to think about why there may be some specific differences as to what we are looking at here. Simply stated, you can easily just look at the scientific names and draw conclusions based on that. But you may want to think about those divisions with the Atlantic and the Pacific clade specifically give rise to correlation with geographic locations. Look at the areas that are circled with the bootstrap values to see how confident the genetic differences are showing those relationships. Think about whose sister taxa to one another. Also think about what type of clades are you seeing. Are they all monophyletic, or can it be suggested that some of the Expenserae family is paraphyletic? After all, note that we see embedded within a lot of our Expenserae, we see other organisms as well, like our huso. Why do you think that's occurring? Is it something with initial misclassification, or did we see different changes in their genetics due to potential hybridization, or even interacting in close home range with one another? I will leave you with this as you look at this particular tree. As always, if you have any questions, don't hesitate to shoot me an email. I'm more than willing to help.